Welcome to BioBits and Bytes. We're always looking into the cutting edge of cancer research, and one area that's incredibly exciting right now is cancer immunotherapy.
Absolutely. It's a rapidly evolving field.
Today, we're really going to dive deep into one specific aspect, neoantigens. We've got some great material here, research from EGA, PMC, clinical views from peer review, even ethical guidance from Laboratorios Rubio.
A good mix of sources.
Right. And our mission today is really to unpack what these neoantigens are, how scientists actually find them, why they're causing such a stir, and importantly, some of the challenges and ethical points we need to think about.
Crucial considerations, definitely.
And thankfully, we have an expert scientist here with us to help navigate all this. So let's jump right in. For anyone new to this, maybe start us off. What exactly is a neoantigen?
Sure. So think about cancer cells. Their DNA, it accumulates errors, mutations that you don't find in normal, healthy cells.
Okay, mistakes in the code.
Exactly. And these mutations can cause the cell to produce proteins that are, well, different, unique to the tumor. Right. Now, when the cell breaks down these unique proteins into smaller pieces, peptides, and displays them on its surface, that's potentially a neoantigen.
Ah, like a little flag saying something's wrong here.
Precisely. A new antigen, neoantigen. It's a signal that the immune system can potentially recognize as foreign, as non-self.
That's a great explanation. So why is this such a big deal for immunotherapy? What makes them promising targets?
Well, it really comes down to specificity. Because these neoantigens come from mutations only in the cancer cells, they offer a way to target just the cancer.
Not the healthy cells.
Exactly. Our immune system is trained, through a process called central tolerance, to generally leave our own healthy tissues alone. Since neoantigens are new, they haven't gone through that tolerance process.
So the immune system is more likely to see them as invaders.
Potentially, yes. The idea is to harness the immune system to attack cells showing these specific flags, hopefully with fewer side effects than treatments that might also hit normal tissues.
Okay, makes sense. So we know they're there, these unique flags. How do scientists actually find them in a patient's tumor? It sounds complex. One source mentioned sequencing and in silico prediction.
Yes, that's the most common approach now. It starts with getting samples, usually both tumor tissue and normal tissue, like blood, from the patient.
Okay.
Then you use next-generation sequencing, NGS. This is basically a very high-throughput way to read the DNA sequences of both samples really quickly and comprehensively.
So you get the full genetic blueprint for both.
Pretty much. And by comparing the tumor DNA sequence to the normal DNA sequence, you can identify the mutations that are specific to the cancer.
Got it. So that gives you a list of differences. What's the in silico part? So the computers are involved.
Right. In silico just means done on a computer or via computer simulation. Once you have that list of tumor-specific mutations, you know which proteins might be altered.
Okay.
But not every mutation leads to a protein fragment that actually gets presented on the cell surface and recognized by the immune system. So we use computational algorithms.
The in silico prediction tools.
Exactly. These algorithms predict which of those potential mutated peptides are most likely to, 1, bind to the patient's immune presentation molecules, and 2, be visible to immune cells.
The paper mentioned some early ones, NetMHC and pickpocket. What were they trying to predict specifically?
They're primarily focused on predicting how well a given peptide will bind to MHC molecules' major histocompatibility complex.
MHC, okay, can you quickly define that? I think I saw HLA mentioned too.
Sure. MHC molecules are proteins on the surface of almost all our cells. Their job is to display peptides, little protein fragments from inside the cell to the outside world, specifically to immune cells called T cells.
Like little display stands.
That's a good analogy. And HLA, human leukocyte antigen, is simply the name for the human version of MHC genes and molecules. So often you'll hear HLA used in the human context.
Got it. HLA is the human MHC.
Right. So algorithms like NetMHC and Pickpocket use data, lots of data from experiments, showing which peptides bind to which HLA types to predict the binding affinity. How strongly will this new mutated peptide likely stick to a patient's specific HLA molecules?
And why is that binding important?
Because if it doesn't bind well, it probably won't be presented effectively on the cell surface, and the T cells won't even get a chance to see it. So predicting strong binding is a crucial first filter.
Okay. Now, another source, the Laboratorios Rubio guide, mentioned something staggering, the sheer amount of genetic data being generated. Billions of genomes by 2025, potentially. How does that flood of data change neoantigen discovery?
Oh, it's transformative. On one hand, it's a massive opportunity. More data means we can train those in silico prediction algorithms to be much, much better.
How so?
By analyzing genome data alongside actual patient immune responses, we can find subtle patterns. Which types of mutations, which peptide features really trigger an immune attack? The algorithms learn and get more accurate picking the winners.
So better predictions. But does it create challenges too?
Absolutely. Managing, storing, and analyzing that volume of data is a huge computational challenge. We need incredibly powerful bioinformatics tools and infrastructure just to handle it, let alone make sense of it. It also raises the stakes on data security and privacy, which we should definitely talk about.
We will. But first, connecting some dots. Peer review listed things like PD-L1, T cells, tumor cells as immunotherapy biomarkers. How do neoantigens fit into that picture with these immune system players?
They're all interconnected. T cells, as we mentioned, are the key immune cells that can recognize and kill cancer cells. They do this by using their T cell receptor, or TCR, to scan the peptides presented by HLA molecule on other cells,
Including tumor cells.
including tumor cells. If a T cell's TCR recognizes a neoantigen peptide presented by an HLA molecule on a tumor cell, it can get activated to attack.
Okay, so neoantigens are the target signals for the T cells. What about PD-L1?
Right, so PD-1 is a protein often found on T cells, and PD-L1 is a protein that tumor cells can put on their surface. When PD-L1 on the tumor binds to PD-1 on the T cell, it acts like a brake.
An off switch for the T cell attack.
Essentially, yes. It tells the T cell, hold off, don't attack. Many cancers use this trick to hide from the immune system. Neoantigen therapies aim to activate T cells against the tumor.
But those activated T cells might still get shut down by PD-L1.
Exactly. That's why combining therapies can be powerful. Use a neoantigen vaccine to generate the T cells, and use a drug that blocks PD-1 or PD-L1, a checkpoint inhibitor, to release the brakes so those T cells can work.
That makes sense. Now, one paper pointed out something interesting.
Yeah.
Just because a peptide binds strongly to HLA doesn't automatically mean you get a good immune response.
Yep.
Why not?
That's a really critical point. Binding to HLA is necessary, but it's not sufficient. The T cell receptor, the TCR, also has to effectively recognize the peptide while it's being presented by the HLA molecule.
So it's a two-part recognition. HLA presenting, TCR seeing.
Precisely. Think of the HLA peptide complex as a single entity that the TCR interacts with. Even if the peptide part is stuck firmly in the HLA groove, the overall shape or the specific amino acids exposed might not engage the TCR strongly enough to trigger a full activation.
So the fit with the T cell itself matters just as much.
Absolutely. It's the quality of that TCR interaction that ultimately drives the T cell response.
And that same paper noted that peptides that do trigger a strong response often have certain types of amino acids, like hydrophobic or aromatic ones, right, where the TCR connects. What does that tell us for designing therapies?
It gives us another layer for prediction and selection. If we know that certain amino acid characteristics at the TCR interface are associated with stronger T cell activation.
You can build that into the algorithms.
Exactly. We can refine the in silico tools to not just predict HLA binding, but also to score how likely the peptide is to effectively engage a TCR based on these properties. It helps prioritize the neoantigens that are most likely to be truly immunogenic.
Okay, so the prediction pipelines are getting smarter. The ESMO guidelines mention these pipelines look beyond just binding. What else do they consider?
Yeah, the modern pipelines integrate a lot more information. For instance, they look at the variant allele fraction, or VAF. Basically, what percentage of the tumor cells actually carry that specific mutation. A mutation found in nearly all tumor cells, a clonal mutation, might be a better target than one only in a small subpopulation.
Okay, that makes sense. What else?
Gene expression is vital. Does the mutated gene actually get turned on and produce the protein? If the gene isn't expressed, you won't get the neoantigen, even if the mutation is there in the DNA. So they often integrate RNA data.
So is the factory even running?
Kind of, yes. They also look at the predicted stability of the peptide HLA complex, how different the mutated peptide is from any similar normal peptides in the body, all trying to get a better picture of which candidates have the best shot.
And Esma also mentioned that neoantigens aren't just from simple DNA typos, but can come from more complex events like frame shifts or gene fusions, even in tumors with fewer mutations overall. Are those particularly interesting?
They really are. These non-standard sources, frame shifts, splice variants, gene fusions, even things called endogenous retro elements that are normally silent, can create peptides that are radically different from anything the immune system has seen before.
Totally novel sequences.
Often, yes. And because they can be so foreign-looking, they sometimes provoke a very strong immune response, even if the tumor doesn't have that many mutations overall. It opens up possibilities for immunotherapy in cancers previously thought less likely to respond.
That's fascinating. Now, the challenges paper contrasted this personalized neoantigen approach with older attempts targeting shared tumor antigens, which it said were often disappointing. Why the difference?
It comes back to that specificity and tolerance issue. Shared antigens are often proteins that are, yes, overexpressed on cancer cells, but might also be present at low levels on some normal cells.
So the immune system might be somewhat tolerant to them already.
Exactly. That can dampen the response. Plus, a therapy targeting a shared antigen only works well in patients whose HLA type can effectively present that specific antigen, neoantigens being derived from unique somatic mutations.
Or truly personalized to that patient's tumor.
Right. The therapy is designed based on their specific mutations and their HLA type, aiming for a much more tailored and potentially potent attack, hopefully avoiding that pre-existing tolerance.
Okay. And we're seeing this move into the clinic.
Yeah.
ESMO detailed ongoing trials using personalized neoantigen vaccines. What kinds of vaccines are being tested?
There's quite a variety of platforms. One common type uses synthetic peptides, lab-made versions of the predicted neoantigens, often mixed with something to boost the immune reaction. Just injecting the peptides.
Essentially, yes. Others use DNA vaccines, where the DNA code for the neoantigens is delivered, getting the patient's own cells to produce them temporarily.
And similarly, RNA vaccines deliver the messenger RNA code. There are also dendritic cell vaccines, which are more complex. You take out the patient's dendritic cells, key antigen-presenting cells, load them with the neoantigens in the lab, and put them back in.
Wow. Several different ways to train the immune system.
Exactly. Each has potential pros and cons being explored in trials.
And you mentioned earlier the idea of combining these vaccines with checkpoint inhibitors.
Yeah.
Why is that combination potentially synergistic?
It's like a 1-2 punch. The vaccine's job is to generate and expand an army of T cells, specifically targeted against the tumor's neoantigens, basically, teaching the immune system what to attack.
Priming the response.
Right. But as we discussed, the tumor can put up defenses, like PD-L1, to shut down those attacking T cells. The checkpoint inhibitor's job is to disable those defenses.
Releasing the brakes.
Precisely. So the vaccine creates the targeted soldiers, and the checkpoint inhibitor clears the way for them to actually fight effectively in the tumor environment. Together, they might achieve a much stronger and more durable anti-cancer effect than either could alone.
That makes a lot of sense strategically. Okay, switching gears a bit, because the Laboratorio's Rubio guide brought up some really critical ethical points around all this genetic data. What are the main ethical hurdles here?
They are significant. Top of the list is probably data privacy and security. Genetic data is inherently sensitive, revealing potential health risks, ancestry, things about relatives. Protecting it from breaches or misuse is absolutely crucial.
And informed consent must be tricky too.
Extremely. Genomics is complex. How do you ensure a patient truly understands what's being done with their DNA, who sees it, the potential implications, good and bad, long-term storage, secondary uses for research? It requires very careful communication.
Right. The guide stressed that truly anonymizing genetic data is really hard, maybe impossible, because it's so unique. How do researchers balance using this vital data for progress with protecting people's privacy?
It's a constant balancing act. There's no single perfect solution. Strong technical safeguards are essential: encryption, secure servers, controlled access. Techniques like pseudonymization, where names are replaced by codes, help, but re-identification isn't impossible.
So what else?
Clear governance is key. Strict rules about who can access data for what purpose, with strong penalties for violations. Transparency with patients about the risks and the safeguards is also vital for trust. And ongoing research into new privacy-preserving computation methods is really important too.
And how do we make sure patients actually understand what they're consenting to, given the complexity? The guide mentions simplifying the information.
Yes, that's paramount. Using plain language, avoiding jargon, visual aids can be very helpful, focusing on what the patient needs to know to make their decision.
Not a full genetics lecture.
Definitely not. It should be a dialogue. plenty of time for questions, maybe follow-up sessions, providing clear written summaries. It's about ensuring that consent is truly informed, not just a signature on a form.
Yeah, absolutely. Finally, that guide also touched on the regulatory side, how laws differ internationally. What challenges does that create for developing these therapies globally?
It creates significant hurdles. Rules around data protection, genetic testing, consent requirements, even the process for approving new therapies like neoantigen vaccines can vary dramatically between, say, the US, Europe, and Asia.
Making international trials complicated.
Vary. And it can affect patient access eventually, too. It requires careful navigation of each region's rules, close collaboration with different regulatory agencies, and ideally efforts towards some international harmonization of standards while still respecting local contexts. It adds another layer of complexity to getting these promising therapies developed and delivered safely and ethically worldwide.
Wow. Okay, well, as everyone can hear, this field of neoantigens in cancer immunotherapy is incredibly dynamic. There's so much potential, but also complexity.
Definitely. A lot moving parts.
We've covered what they are, how they're found, using some pretty amazing tech, and importantly, the ethical and regulatory landscape that has to evolve alongside the science. That journey, from finding one mutation to hopefully directing the immune system against it, it's intricate.
But the potential payoff for patients is huge.
Immense. It really highlights the forefront of cancer research, sequencing, bioinformatics, immunology, all coming together. We really hope this deep dive gave you all a clear understanding of this exciting area, maybe sparked some curiosity too.
Hopefully so.
A huge thank you to our expert for breaking down such complex topics for us today. Really appreciate your insights.
My pleasure. Happy to discuss it.
And thank you all for tuning in to BioBits and Bytes.